In this presentation, we will occupy ourselves with unifying wiring principles pertaining different scales and species. The organization of the brain of different mammals is characterized by different levels, different scales of organization, that is, its large-scale wiring or its cytological composition. And recent endeavors have mapped in detail these different levels of architecture. For instance, certain advancements in the field of connectomics have unraveled in detail the intricate wiring of mammalian brains, including the monkey, the human, and the mouse brain. Moreover, projects like the Big Brain Project have mapped in detail the cytological heterogeneity of, in that case, the human brain. Thus, we have at our disposal a wealth of unprecedented data, but certain key challenges remain. Namely, given that we have this wealth of data, what is a parsimonious set of principles that can describe in a numerologically informed way these different scales of organization and uncover potential unifying threads that bind these observations across different species? So this question constitutes the motive of the study that I will elaborate on. The starting point is thus to have these raw observations and aim to distill in a parsimonious manner key components that help us understand the relation between these two different scales across different species. Thus, we have a transition from observations to principles, and the explanatory depth with respect to the scientific object at hand, that is the mammalian cortex, is enhanced when we transition from left to right, as we observe in this diagram. Our starting point is the cytological composition of the mammalian cortex. Studies of decades have uncovered that the cytological composition of mammalian cortices is heterogeneous. That is, we have to do with spatially ordered changes in terms of cytoarchitecture. And these changes can be quantified in different species by using the so-called neural density. This simply means, given a cubic millimeter of cortex, we count how many cells inhabit this cubic millimeter. And if we do that in the macaque monkey, we see that we have these spatially ordered gradients of cytological heterogeneity. For instance, the entorhinal cortex exhibits very low density, but as soon as we start transitioning towards primary visual areas, and we end up in B1, we see that the neural density becomes higher and higher. So we can quantify with neural density the cytological heterogeneity that manifests as spatially ordered changes across the cortical sheet of different mammals. At the level of connectivity, studies have shown that there is also an heterogeneity that pertains to the wiring of the brain. From a topological standpoint, certain areas are more central than other brain regions, and these central areas are tagged as hubs or core. We have a highly interconnected set of areas. In contrast, the periphery refers to areas that are not heavily interconnected with the rest of the brain. So we have to do with heterogeneity, but now at the level of connectivity, at the topological level. The question is, if and to what extent cytology and topology relate to each other across different species. To this end, We map cytological gradients in different mammals. Specifically, we map these gradients in the mouse, the cat, the macaque monkey, and the marmoset monkey. So we have a variety of species, carnivores, rodents, Old World and New World monkeys at our disposal. By doing so, the next step is to use the extensive wiring diagrams that exist for these species and uncover the more central regions in each one of these species. That is, the goal is to uncover the core-periphery structure in these different mammalian species. And we did so, and the core is denoted here with this color and the periphery with this color. And once we have mapped these topological features in different species, the goal now is to relate them to the cytological composition of the areas. And as we see here, in mice and marmoset monkeys, topology does not lead to a statistically significant segregation in terms of cytology. That is, there is no statistical difference between core and periphery areas in terms of the neuronal density, the cytoarchitectonic status, that is, of these areas. On the contrary, in cats and macaque monkeys, we observe a cytological segregation of core and periphery areas. And the segregation entails that central areas, the core areas, have a lower neural density when compared to periphery areas. That is, the core areas encompass the parts of the cortex that are less densely populated, less cytologically differentiated with this. So we have to do with a species-specific manifestation of a principle that relates topology and cytology in mammals. What could be the ramifications of such species-specific relations of cytology and connectivity? To this end, we build on observations in the macaque monkey, and these observations relate the cytological status of areas, specifically the prefrontal cortex of the monkey that ranges from low to high density areas. And the study relates these neural density variations to myelin content of areas. And as it's here depicted, the less cytologically dense areas also exhibit low myelin content. And this stepwise gradual increase in cytological composition, neural density, is accompanied also by an increase in the overall myelin content. So that means more myelin is equal to higher levels of neural density. And from a functional standpoint, this relation helps us understand the degree of plasticity of these areas, because these gradients of cytology and myelination are also entailing changes in terms of the plasticity of each region. For instance, high plasticity is accompanied by low neuronal density. In other words, the more myelinated an area is, and the more neurally dense an area is, the lower the plasticity of that area is. With these observations now, we can have a potential functional ramification of the phenomena that we observe. That is, the displacement of the core that we have observed in our own studies is now interpreted in terms of plasticity. So we have to do with an increased segregation of core and periphery in terms of cytology as we transition from mice to marmoset monkeys, cats, and macaque monkeys. And this cytological segregation now also entails, in light of the findings that we have previously iterated, that the network core in the cat and the macaque monkey also encompasses the more plastic parts of the cortices of these species. In summary, we have a species-specific alignment of the cytological and topological gradients in the mammalian cortex, and when this relation is pronounced, we have to do with the more central, less dense principle. This displacement of the network core in the mammalian spectrum indicates that all both monkeys and carnivores may exhibit a network core that is heavily interconnected areas that also encompasses the more flexible parts of their cerebral cortex. And this might have implications for flexibility and adaptability into a niche, for instance. And with that, I would like to thank you, and I hope you found something of interest for your own endeavors.